Hi guys, welcome to my presentation. My name is Joyce Liu and I am a student at Emory studying neuroscience and behavioral biology. My research looked at fear responses to predator odor in mice from a pharmacological and neuroanatomical perspective in order to understand the contributions of dopamine and norepinephrine to fear and stress-related behavior. We were specifically interested in looking at fear responses because it is an innate emotional response that's critical for survival, and its dysregulation is implicated in many psychiatric diseases. Although norepinephrine has long been known to play a role in stress responses, more investigation is needed in order to understand its exact contribution. Unfortunately, we have a model for studying this through the model of Toxoplasma infection. Toxoplasma gondii infection in rodents abolishes innate fear of predator odor, and it has been hypothesized that this is accomplished through the suppression of dopamine beta hydroxylase. Um, so this is also known as DBH. So DBH is an enzyme that converts dopamine to norepinephrine, and its suppression then results in a buildup of dopamine and a reduction in norepinephrine. Therefore, the goal of the study was to determine the effects of genetic and pharmacological disruption of DBH on behavioral responses and neuronal activity following exposure to predator odor. So we used two types of genotypes of mice in this experiment. DBH knockout mice, which completely lack norepinephrine and have increased dopamine, and also their norepinephrine competent littermates. So in the first experiment, the two genotypes were compared to each other, and in the second experiment, norepinephrine competent mice were treated with the DBH inhibitor napikostat and compared with the same mice treated with a saline vehicle. So in the behavioral analysis, individual mice were removed from their home cages and placed in a new cage with a cotton nestlet soaked in a neutral novel odor. And the first 10 minutes of behavior were scored for digging, grooming, and nestlet contact time. After 90 minutes, we assessed if the nestlet had been shredded and turned into a nest and if the animal was asleep. The same analysis was repeated for exposure to a cotton nestlet soaked in predator odor. 90 minutes after predator odor exposure, the knockout mice and control mice were perfused for immunohistochemistry of CFOS and norepinephrine transporter to see the overlap of neuron activity and norepinephrine transmission. So from our first experiment, comparing DBH knockout mice with norepinephrine competent controls, we found that knockout mice have significantly increased grooming and decreased digging regardless of the odor presented. Nestlet contact didn't really vary by genotype, but it did by odorant. So while the knockout mice were less likely to shred the nestlet soaked with the neutral odor, they were just as likely as the controls to be asleep after 90 minutes. And shockingly, in the presence of predator odor, while the control mice didn't shred the nestlet or fall asleep, the DBH knockout mice were more likely to form a nest and fall asleep. So from our immunohistochemistry of these animals, we found that the knockout mice had reduced neuronal activity in several areas implicated in stress responses to predator odor. Specifically, they showed reduced activity in many regions that received noradrenergic innervation from the locus coeruleus. Abnormalities in neuron activity throughout this noradrenergic circuit may explain the behavioral differences between the knockout and control mice. In our second experiment, we performed the same behavioral analysis on norepinephrine competent mice treated with the DBH inhibitor napikostat. We found that the DBH knockout mice behavior in the presence of predator odor could be recapitulated with this pharmacological treatment, which supports the idea that DBH has a function in innate fear, and disruption of norepinephrine and dopamine transmission may alter the recruitment of neural circuits that organize behavioral responses to predator threat. Future experiments targeting the specific transmission of each catecholamine will help disentangle the relative contributions of each to fear responses. So that's my presentation. I'd like to give a special thank you to my mentor, Danny Lesberg, and my PI, Dr. David Weinschenker, as well as thank you to the funding from the Emory Med School's Human Genetics Department and the Summer Undergraduate Research Experience Program for making this project possible. And thank you all so much for listening to my presentation.